Many studies of the ecological effects of environmental change focus on a single aspect of change. For example, early experiments at Cedar Creek examined effects of fire, nitrogen inputs, or biodiversity in isolation. But humans are changing the environment in lots of different ways at once, and the combined effects of these different global changes might differ from the sum of their individual effects. Recognition of this complexity led Cedar Creek LTER researchers to establish some of the first experiments in the world to examine how diverse global environmental changes might interact to affect communities and ecosystems. Now more than 20 years old, the BioCON experiment, short for biodiversity, CO2, and nitrogen, is one of the longest-running experiments in the world to increase carbon dioxide in an outdoor setting. Several hundred plots within the CO2 rings are planted with different numbers of species, and half of the plots receive added nitrogen, so researchers can explore whether the ecological effects of elevated atmospheric carbon dioxide depend on soil nitrogen supply and the diversity of plants in the community. About a decade ago, 2 additional treatments were added to a subset of the BioCON experimental plots. Temporary rainout shelters erected during some summer storms reduce annual rainfall by about 30%, and a warming treatment uses infrared heaters and soil heating rods to warm plots by about 2.5 degrees. In the early years of the BioCON experiment, plants receiving added nitrogen grew no more under elevated CO2 than plants that did not receive nitrogen. However, after about 3 years, plots receiving nitrogen produced more biomass when growing under an elevated CO2 atmosphere. These results and others highlight the importance of long-term experiments, as short-term results would have led researchers to the wrong conclusion about how nitrogen and CO2 interact to affect plant growth. More recently, BioCON researchers have seen similar interactions between water and CO2. Plants receiving higher levels of rainfall are able to grow more with elevated CO2 than those receiving lower levels of rainfall. These findings add to growing evidence that plants are often simultaneously limited by multiple different resources. Plant species in BioCON come from diverse functional types: warm season C4 grasses, cool season C3 grasses, legumes, and other herbaceous plants. When more species and functional types are growing together in a plot, biomass increases more with elevated CO2 and added nitrogen, likely because collectively, mixtures of species are able to acquire more of all kinds of resources than any of them would growing alone. In other Cedar Creek experiments, LTER researchers have been overlaying new treatments to further understand how biodiversity interacts with various global change factors. New drought and warming experiments are addressing a decades-old mystery about whether biodiversity makes communities more resistant to extreme climate events. During one of the worst droughts in the past century, plots that had lower plant diversity because of being fertilized with nitrogen showed large reductions in productivity compared to unfertilized plots. Were fertilized communities more vulnerable to drought because they contained fewer species and were poorly buffered against drought? Or did species that flourished with added nitrogen have characteristics that made them suffer with drought? New LTER experiments at Cedar Creek are specifically designed to tease apart this complexity. For example, research in the original Big Biodiversity experiment is examining whether more diverse communities show less fluctuation in productivity with experimental drought. The rich suite of long-term experiments at Cedar Creek LTER that consider multiple global changes simultaneously allows researchers at Cedar Creek and around the world to mechanistically explore how these changes are interacting to alter diverse aspects of ecological systems.